Hi everybody, thank you for supporting another seminar series. This one's for year 11 and 12 biology. And it'll be run in much the same vein as the last one. A 1-hour recorded seminar, tutorial questions, how-to videos, or explanatory videos uploaded each week, as well as some other ones as well. So the schedule will be, this week will be cells, bacteria and viruses. Saturday the 27th will be hierarchy of living systems, animal anatomy and physiology, genetics, inheritance and evolution. And my favorite one is human systems biology, plant animal and physiology, plant anatomy and physiology, stem cells and gene therapy, mitosis and meiosis, and looking at genetic diseases, genes and proteins, cancer and telomeres, and I've decided to do an extra one on immunity and the molecular biology evolution, which is probably the one I'm looking forward to the most. So today's session will look at cells, bacteria and viruses. Quite a topical one. It covers a big chunk of the curriculum. So the cells are the smallest living unit. They date back 4 billion years. I think the first living bacterial cell, fossilized bacterial cells have been found. And we named them after what we saw under a microscope. So, plant versus animal cell. Plant cell is quite different. It will seem a little bit basic for those who are doing year 11, 12, a little bit of uni biology, but I like to rehash the basics. I'm sure you guys are all aware by now. So plant cells look like, they don't all look like this. This is the classic representation. Animal cell is, depending on what the cell is represented as a circle. They both have a nucleus, but the key differences, I know that says cell wall, but it's supposed to say cell membrane. Plants have a cell wall, animal cells have a cell membrane. You can see it now. Cell wall and cell membrane. Plants also have these chloroplasts. So if we remember back to photosynthesis, and reshuffling my desk, we know that chloroplasts are the things that absorb the wavelengths of light. They can be green, red, and yellow, I think. But the most common one is green, and they live in the leaves, hence why the leaves are green. So big differences are those two. And they are, as most things, related to structure and function of the cells themselves. Reshare this. So prokaryotic and eukaryotic. Again, this will be a quick rehash for most of you. So eukaryotic. Prokaryotic are simple. unicellular, and no membrane-bound organelle. So bacterial cells do not have mitochondria. Or, sorry, yeah, or chloroplasts. So DNA in eukaryotic cells is bound within the nucleus. Within prokaryotic cells, it's just floating as a single molecule, all within plasmids. And if you go to uni, you'll learn all about plasmids, and that these plasmids are what we use to take genes from one plant, one organism, and stick a new gene in, and then transplant that in. That's the basics of genetic modification. I won't go too deep into that. Eukaryotic are the opposite. So, the exact opposite of prokaryotic. Good way to remember that: eukaryotic is so complex, multicellular. Eukaryotic, as prokaryotic is simple. And so, just like we discussed there, so you can see on this picture that they simply have one big molecule for all their nuclear material. They've got the cytosol and these plasmids of circular DNA. They also have these flagellum. If you put bacteria into a solution, you can watch them swim around under a microscope. So again, we just discussed that. So this is probably one of the big differences between what you learn in 7 to 10 and what you're expected to learn in year 11 and 12. So the knowledge of how everything comes together in itself. Hopefully you can remember that screen, or you've got this. I'll send the slides out after this. Got you that this morning, do apologize. You have the cell membrane. In the middle we have the nucleus. Surrounding the nucleus, and I'm just going to draw it on this side for ease of. We have the endoplasmic reticulum. So, and on the endoplasmic reticulum, we have ribosomes. Now, it's called the rough endoplasmic reticulum because it has all of these ribosomes on it. Now, all of the genetic information stored in the nucleus in these chromosomes. If I took the hard drive off my motherboard, my computer there, and set it over there, it would be nothing but a set of instructions. And that's what this is. Set of instructions. And inside here, we have the machinery. I'm not going to go into this because we will cover it in detail later on. But we have machinery that unpacks this DNA and transports it over here. The ribosomes, based on the genetic code, will put all of this together, and what is produced from the nucleus is proteins. So the ribosomes will put the proteins together, and as they go down this endoplasmic reticulum, they will fold up and be ready to be used. And then we have, so I'm just going to write ER and ribosomes. Then we have the Golgi apparatus on the end. And what happens here is these proteins, depending on where they're going, this is still, I should say this is still inside the cell. I think I do that very well. So that's our nice long cell. The Golgi apparatus packages the proteins up into vesicles. Those vesicles either get sent into the cell to do something, or they get sent to here, where they fuse with the cell membrane and their contents is ejected out of the cell. So what we call endocytosis. We also have what's called mitochondria, and they often look like a piece of bakery bread. And they are what's called the powerhouse of the cell. They produce energy. They produce the energy required. Got a few others, and throughout the cell, I'm just going to wreck my beautiful drawing. We have, it's called the cytoskeleton. It runs like this. And what it does is, so when this protein is told, hey, I need to go out, it will follow this, and it will follow that, and then it will head out. If it goes, oh, I need to be back here, it will follow this cytoskeleton to where it needs to go. It also allows the cell to be firm and not too squishy, but not too hard. And it's made up of these microfilaments, intermediate filaments, and microtubules. And these are important when our cell begins to divide, and I will go over why in later weeks when we look at cell division. Everybody's favorite topic. So the nucleus, and you see those nuclear pores on the outside. That is for when the information needs to get down into the endoplasmic reticulum, into the ribosome. So the condensed chromatin are all of your chromosomes. endoplasmic reticulum. You can see there the proteins go down the maze or the gauntlet, whatever you want to call it, and they're added to by the ribosomes, and then they leave through these nuclear pores. So you've got the smooth endoplasmic reticulum and the rough endoplasmic reticulum. So the ribosomes, based on the genetic code which is provided for by messenger RNA, so from the nucleus, we get messenger RNA, which carries a code to the ribosomes, and they build, the mRNA goes in this way and is read. I mean, this is a very simplistic representation. And the protein is built coming this way. So as the mRNA gets shorter, the protein gets longer, or the amino acid chain, peptide chain, whatever you want to call it. I'll go through that again in more detail in the coming weeks. This is just a rehash. So bacterial cells, they are prokaryotic, and the nuclear material is either circular or is in one big chunk or one molecule. So we break up our bacteria into gram-negative and gram-positive. So gram-positive broken up into gram-positive and gram-negative. Gram-positive, they have this molecule called peptidoglycan in their cell wall, and when you stain it with this crystal violet stain, then it looks purple under a microscope. So examples, Staphylococcus aureus, or golden staph. If you want to gross yourself out, have a look at what happens to the skin when you have a golden staph infection. You'll see why it's called golden staph. It lives in the nose and on the skin, and it's really one of our biggest public health issues is antibiotic resistant Staphylococcus aureus. Basically meaning that we can't, oftentimes the only method of treatment is to physically cut out the infection because we can't treat it with antibiotics because it's become resistant. So antibiotic resistance is caused by overuse of antibiotics. It is a natural thing because almost all of our antibiotics that we use are natural. They have been sourced from the natural world. We do have some more synthetic ones coming through now. But there is natural resistance out there to these antibiotics. Most of them come from other bacteria. So very quickly when we start ramping up an antibiotics use, we start to see resistance. So diphtheria is tetanus. So the reason you should get your tetanus shot, when they say, is because your body will quite literally lock up and you won't be able to move. And anthrax. Anthrax is found in sheep, I'm pretty sure, and is a very, very potent biological weapon. So gram-negative, they have many times higher levels of antibiotic resistant than gram-positive do. So STIs, there are STIs out there now that they cannot cure. So as you get older, just keep that in mind. 100% resistant, we cannot treat it. So they're much harder, but when you put the crystal violet stain on, they don't retain it. When you wash them off, they don't retain the stain. They are the hardy ones and the really worrisome ones. So I think I just spoke a bit about antibiotic resistance, but bacteria can do some cool and wacky, wonderful things. So it only takes one bacterial cell that's resistant to make an entire community of bacteria resistant. So once you, just in case you didn't know, these are bacterial cells. Bacterial cells can create a tube between themselves. Looks like a dumbbell or something, right? But a tube between themselves, and they have these plasmids that we learned about before. And now these are where the site of where things that need to be produced quickly are on plasmids. So there you go, hey, I've got this antibiotic resistance gene. Do you want it? And this guy goes, yeah. And then he gives it to him. Now they've both got it. And then you start putting selection pressure and all the other bacteria die. So we use an antibiotic. That's why they say you have to finish a course of antibiotics. If you don't, and there are bacteria that are resistant and there are any left, that one will just pass it on to the next and you'll never get rid of the infection. So bacteria are very, very good at adapting very, very quickly. So pathogenic bacteria, it's all in the name, they're pathogens, they have the ability to cause disease. The reason that's the best public health campaign there is for people to stay healthy. When you're healthy, you can fight off just about anything. And I say just about anything, there's certain things where it doesn't matter. But for all intents and purposes, if you keep yourself healthy, you can fight off just about anything that will come your way. Strep throat is an infection of streptococcus in the back of the throat. Urinary tract infections are as uncomfortable as they sound. So they're caused by E. coli. Meningitis is bacterial infection of spinal fluid and brain stem, which is very nasty. Anyone has to deal with that. I'm sure would rather forget it. Cellulitis, which is, it's quite literally, your skin becomes infected. Now, most people, when they're healthy or when they've got intact skin, you get a cut on your arm and there's bacteria there because you haven't showered or you came into contact with someone or you didn't wash your hands after you blew your nose, because that's where all the bad bacteria live in a lot of the time in your body, is in your nose. All of a sudden that's been transferred into that cut and you are in a world of pain. So we have indirect, direct, and vector associated transmission. So examples include coming into contact with bodily fluids, skin-to-skin contact, and coming into contact with contaminated surfaces. A couple more are mosquitoes, ticks, we don't really have Lyme disease. In Australia, there's some people who would agree or disagree with that. And airborne. There's not many things that are airborne. Measles, which is about 50 times as infectious as coronavirus. If you can get measles by walking into a room where someone, even a few hours before, had measles. So that's why it's so dangerous and they're so quick to get on top of that, and it's obviously to get people to have the vaccine. So antibiotics, let's sit down. I don't think there's much more board stuff. So antibiotics are They're designed to kill bacteria. There's also antibiotics for fungal infections, but they're called antifungals. Antivirus is antivirals. But we know that bacteria have been growing and evolving for a very, very long time. And in that time, they've had to fight each other off. So logically, the best place to find antibiotics is to go to the source. So the guy, I can't remember his name now. Why did I not write that down? But he left his experiments with mould. He was doing some kind of bacterial experiment. And he came back and he saw mould spores on his plates, and he thought, oh, that's not good. But what he also saw was a ring around the outside that was completely free from bacterial growth. And this was in a long time ago. And they couldn't actually extract what they couldn't actually extract what was doing it. But it wasn't until about 50 years later that they could. An Australian guy, actually. I can't remember his name, Andrew Thomas. And he was able to extract that, and that's where we get penicillin from. But there are stories and medical literature from about 5 years after we introduced antibiotics that were showing signs of antibiotic resistance. And we're now in the antibiotic resistant age, or where it's thought that previously easily dealt with infections will kill thousands, if not millions of people each year. Because we don't have any new drugs to treat them, the bacteria are becoming resistant to the antibiotics significantly faster than we can produce. Now, as I said before, to scare you all, and that's actually true, I did say to scare you, that you're 100% resistant to bacteria. So viruses, and I'll put out a video explaining what living and non-living is in this context. They require a host to spread and to survive and to replicate. Without that host, they would not be alive. That's the big reason why they're not considered to be alive. The virus doesn't actually kill the host. In most cases, it is, so with coronavirus, it is a case of the body mounts such a massive immune response and an overreaction that the person drowns from the inside out when they do that. So that's not the virus doing anything. It's causing it, but nothing the virus is doing is actively killing you or injuring you. So they use the cellular machinery to create copies of themselves. Enveloped viruses, coronavirus is an enveloped virus. It is a process called budding, where it quite literally just steals a piece of the cell membrane and then escapes through the cell's defenses. And the body doesn't immediately recognize it as a threat because from the outside it looks the same. So non-enveloped viruses, HIV, which causes AIDS, is a non-enveloped virus. And they just use the cells to replicate themselves until they've replicated so many times that the cells explode and they just travel to the next cell, and they keep doing that. So COVID-19 is caused by coronaviruses. About 30% of the seasonal cold infections come from coronaviruses. We've got SARS and MERS, which both coronavirus, and both were supposed to be pandemics, but thankfully both SARS and MERS just fell off the face of the planet. They mutated themselves out of existence. So SARS had a 10% fatality rate. Coronavirus has like 0.4. I think it's the latest figures. MERS had a 30% fatality rate. So 1 out of every 3 people that got it died. But thankfully, they don't exist anymore. So it causes cytokine storms in the lungs, which drowns the person. The release of fluid is intense. And I mean, among other things as well, but that's the main one. So I'll give a supplementary video on COVID-19. Won't go through it here, it's a whole other topic in itself. So Ebola is a hemorrhagic fever. It's got a 50% fatality rate. I think realistically it's probably closer to 30 from what I was reading today. So it's endemic to certain parts of Africa, Democratic Republic of Congo, Sudan, South Sudan, places like that. And it's significantly more times more deadly than coronavirus. But the death that it causes such a panic and terror that it doesn't really have the potential to become a global pandemic. If you told people that coronavirus, 99 point whatever people will recover without problems, people aren't worried about it. You have a virus that causes death through internal bleeding and going mad, then you get people start to notice and they start to get very worried. So HPV, the human papillomavirus, is a class of viruses, not just one virus. So generalized as human papillomavirus. So unfortunately, almost all cervical cancer cases are associated with HPV. And they recently changed the name from cervical cancer vaccine back to HPV vaccine, I think, because it was a little bit misleading. People were like, well, I can't get cervical cancer if I do this, if I get this vaccine, which just wasn't true. But almost in the high 90% of cases are caused by this virus. So genital warts and cancer. So when I was at the TIA Translational Research Institute many years ago now, and that's where they came up with it. And this trial was so successful for this vaccine that they had to stop it. In a vaccine trial, you have to give 50% of people salt water and 50% of people the vaccine. So you can see what doing nothing does and what doing something does. And it reduced the cervical cancer rates in those young people to virtually nothing. And they had to bring everybody back who'd been through the trial, check which vial they've been given, and then give those people that had those people that have been given the salt water, and then actually give them the vaccination, because it was not ethical to continue knowing how effective it was and knowing that there's people out there thinking they were protected, but they actually weren't. So all viruses are bad. There's hundreds of harmless viruses that get into us that the body can get rid of really quickly or when they enter. And I say all, again, for the scope of this course, that it's the body's response that usually causes the issue. So fever is the body's attempt to bake the virus out. The virus works best at 37 degrees. If the body ramps the temperature up to 40, then it has a better chance of killing off the virus. Anyone that's had a temperature over 40 will understand just how uncomfortable that is. So when people get a slight temperature and they reach immediately for Panadol, I mean, in kids, yeah, fine, give them Panadol when they have a temperature. But when adults get a very slight temperature and then you take Panadol, they're actually prolonging the pain that they're in. If your temperature is 40 degrees, then you need to take Panadol. I had the flu when I came here, 2019, and I was hallucinating in the bottom of the shower for like 3 hours. My temperature was so high. I don't really remember much of that 3-day period, just that it felt like my whole body was on fire. So plant viruses. Plants can't walk. They can't see a threat and walk away. So their viral response and immune response is really, really complex in comparison to ours. They can quite literally calcify parts of the plant to trap something in there. And there are a lot more physical defenses than we do. So tobacco mosaic virus is an example. Tomato mosaic virus. Lots of plant viruses that cause disease in plants, and that's why we have to be so careful at the borders. Those of us who are old enough to remember the old no fruit beyond these points to stop the spread of fruit fly between Queensland and New South Wales. So parasites, malaria, it's caused by a protozoa. Fungi, so yeast, candida, causes thrush and other yeast infections. Antifungal, antiparasitic, or antiviral drugs to get rid of those. Okay, so thank you. I don't even know how long that took. 40 minutes? Okay, I'll upload a few extra videos and make my next presentation a bit longer and more in depth. So, explain the difference between non and non-living, living and non-living in the context of viruses. I'll upload a video about that. And explain what makes human cells eukaryotic. So, I thank you for watching, and I'll make the next one a bit longer and I'll upload a few extra videos. So, thank you.